Okay, so it looks like we are ready to go. Um, so this is going to be like my third time recording this, just because the other two videos were about an hour, an extra hour long, and I kind of want to condense this even further. You guys already had a part one that was like an hour, 10 minutes. So I'm going to try to condense this even more. Since this is review for the most part, there are some things I do want to highlight and kind of expand on just slightly further. But just know that this is chemistry mainly. And the first half of the chapter two is 111A, or the first semester chemistry, and this is now going into the second semester of chemistry. So you will need to review over your delta G's, your P delta V equals work. You'll have to kind of refamiliarize yourself with those concepts. I'm not going to touch too heavily on it, but just note that some of the practice problems at the end of the chapter does ask you to solve for delta G using equations and everything. Now, in regards to our exam or any type of assessment, in if it's a quiz, then obviously you guys will need to know the equations. But if it's on the exam, most likely I will be providing it for you guys, the equations themselves. It's up to you to kind of know where to plug things in and how to digest information given to you in the problem, just like you did second semester chemistry. So, without further ado, let's get started. I don't want this to be another hour and a half long video, so we're going to go straight into it. So we stopped at in the last video at the energy of the cell. We've talked about the first half, which is the macromolecules, the lipids, the proteins, the carbohydrates, and how to recognize them. And now we're going to go into where does energy come from? What is energy and how does our cell essentially where do we get it and what is it used for? Now, you guys can read the list right here, but in so doing, we know that we're going to need to digest food. We know that we got to break down things through a catabolic process to have building blocks to be able to create things inside our cells. And it takes a lot of energy to break things and build things. And so we need help. Our cells absolutely needs help. It cannot do it on its own. And so we're going to enlist the aid of enzymes to help catalyze our biochemical processing. Now, you guys know with enzymes are like tools. We want to reuse them. We don't want to have to recreate them every single time we run a chemical reaction. And we know when we use a proper tool for any type of job, say that you need to screw in some things into the wall, you need to hammer some things, you need to transport some things from one place to another. If you have the right tool, it's going to require less energy on your part to perform that action. Say that you need to move, right? If you need to move from, your current apartment to a new place, say that, you purchase or renting from a condo or something, you got to move all your things. Now, you don't want to carry the boxes one by one from one place to another. It requires way too much energy to transport that. However, if you were able to, say, rent a U-Haul truck, load up everything inside the U-Haul truck, and then drive the U-Haul truck to your new destination, it's less energy on your part relative to you carrying it one by one. So, kind of have that analogy while we are studying enzymes. Enzymes do the exact same thing in concept. Okay. And so these are just images of different types of, I want to say, how do I say this? These are images of different types of things, organelles, cells, viruses. It's a lot that we are looking at. And you can see the scaling difference. And if you were to take a look at the scaling difference, look at this. This is 20 nanometers. Now, remember, when we're talking about eukaryotic and prokaryotic cells, we're dealing in the measurement of micrometers. Nanometers is much, much, much smaller than that. So instinctively, you should already be looking at A and be like, hey, this is way smaller than a cell. It must have something to do with a piece within the cell. It's an organelle or it's a molecule that is found within the cell itself. It's not a cell. Okay. So these again are some things that you should be looking out for. You can see at the very end right here, the succulent plant right here, it's 20 millimeters big. You know this is a huge cell. You know that this piece right here is half a millimeter. So again, it is a fairly big thing. So just pay attention to that size difference. Pay attention to what you are looking at and what can we digest from it. Remember, a picture is worth 1,000 words. And in science, we use a lot of graphs, we use a lot of imagery to kind of visually tell our reader or you guys more than if we were to write it in an essay format. Okay, so cells metabolize using enzymes. Again, we have starting pieces called substrates. It binds to an active site of an enzyme, and then the enzyme will run a catalytic reaction or help aid in a catalytic reaction, which then entails a product that's formed. Now, a lot of the times when we are running through enzymatic reactions, and let me find my pen here, we are not just going from a substrate to a product right away. Sometimes you're going to run through a multi-step catalytic reaction where we need help from multiple enzymes. There we go. Oh, what's going on? Why? Okay, so there we go. Okay. We have multiple catalytic enzymes right here, multiple enzymes. So we start with molecule A. But remember, molecule A is just a starting material. Maybe we need to convert it into something totally different or add things to it. And so we're going to get help from multiple enzymes. So from A to B, that's one enzyme catalytic reaction, B to C, C to D, D to E, and E to F. So essentially, to get from A all the way to F, you need help, a lot of help. Think of it like a processing plant, right? You have raw goods, you have raw metal, you need to make a soda can. How do you do that? Well, you gotta get a slab of metal, you gotta flatten it to aluminum foil size, and then you gotta fold it, and then you gotta, seal it, install the inner lining, the wax lining inside the can, and then fill it up with soda. So essentially, these are step-by-step process. Okay. Now, remember, as we're digesting things, as we're breaking molecules down, this is what we call a catabolic pathway or catabolic reaction. Catabolic means breaking down of. Uh, and energy is usually conserved throughout, whether it's potential energy, kinetic energy, doesn't matter. It should be conserved. But however, we live in a real world scenario. In an ideal scenario, the total energy gets conserved from reactant to product. In a real world, from reactant or substrate to product, we have energy transferred, but a lot of that excess energy will get lost as heat. And so the heat is what gets released into the environment, which also kind of adds to other chemical pathways through the help of thermal energy or thermal motion of the outside molecule. So this step right here is still important. We're still going to utilize some of that heat energy elsewhere. Might not be direct, it might be an indirect, but it still is useful. Okay. Now, when we have a breaking down and we have our building blocks now we can use those building blocks and start piecing them together. And this is what we call an anabolic pathway. Anabolic to build up. So it takes a lot of energy to go from broken pieces to fully formed pieces. So we need help. We need to install energy. We need to introduce or inject energy into the system in order to build. Okay. So heat energy from the cell. Again, this is just an example of spontaneity. Now, Ken be talked a lot about spontaneity. You guys should review it if you're still kind of struggling. But in essence, to keep things in an orderly manner, you need to put energy in. When you're cleaning up your room, you spend a lot of energy. You're trying to put things in a nice, neat pile. But over time, as you are doing your daily activities and you're not really keeping in mind of your room, it eventually becomes slightly messier than when you originally cleaned it. That's why we have like a spring cleaning day or a winter cleaning day, whatever day you designate as your cleaning day. But it is a day that you're mentally telling yourself, okay, I'm going to spend a lot of energy to clean, to go from a disorderly state to an orderly state. So energy required to go into orderly state. Disorderly state is very spontaneous. It just happens without any excess input in energy. Okay. So as we can see, before the cell begins to put things together, where's my mouse? Okay. It has a lot of disorganization throughout. Now remember, to put things together requires energy. And as we know, through a catabolic or an anabolic pathway, heat is going to be released or absorbed, depending. However, as we are performing an orderly positioning right here in this cell, and I'm drawing with my mouse now, okay, where we're creating these organized molecules, we've increased the order within the cell itself. It requires energy. It requires, a certain amount of heat being released. And because the heat's being released, it's going into the surrounding. Now, what's in the surrounding is other molecules. Could be air molecules, could be uh, vitamins, minerals, whatever's around that cell. It's the heat is going to escape. And as you guys know, as you introduce more energy outside into the surrounding, it gets hotter, it gets warmer. And so the molecules outside that cell is going to absorb that heat energy and it's going to start moving faster, increasing the disorder of the surrounding. So the system, the cell itself is increasing in order, increasing in organization, and outside the cell, it becomes more disorganized or more disorderly. Okay. So here is just a depiction of energy conversion. You have potential energy from raising a brick. If you drop a brick, you hear the sound when it lands, and you see it go from a still position to a motion. Okay. So potential energy is when everything's standing still. If you let go, it just happens. Energy is converted from a potential to a kinetic. Kinetic means movement, and then you hear it. So, molecules. All the energy is found within the cell. It's not in the cell, in the bonds themselves. Remember, you have two nucleus, positively charged, repelling, trying to repel, but it's being locked together through a covalent bond. So you're going to have vibration, you're going to have stretching between the nuclei and the covalent electrons. You're also going to have wobbling, flipping, bending. That's all potential energy locked into that cell or into that molecule. Now, if we cut that bond, what's going to happen? It's going to spring out. It's going to release all that stored potential energy out into the surrounding. Okay. And then a battery and sunlight, you guys hopefully get the idea. Okay. Now, cells obtaining energy through oxy-redox reaction. We kind of talked about this in chapter 1 when we were doing our practice problem. How we get energy is through digestion of food for us. Now, how do we digest food? Oxy-redox reaction. All biochemical pathways, all organic pathways, is all reactions occur when one electron is being transferred or multiple electrons are being transferred from one molecule to the other. Oxidation, reduction, oil rig. Okay. Oxidation is loss of electrons. Reduction is gain of electrons. Okay. So we can see right here, if we take a look, we have our water, we have our oxygen. And we know that through water in photosynthesis, we're going to strip away the hydrogens. Now, why is that important? Because the hydrogens are important for the hydrogen ion concentration. We need to build that gradient, right? So that diffusion later can happen from high concentration to low concentration, turning the ATP synthase for the most part. Okay. So you have some gradient moving. So water is responsible for providing hydrogen ions, but it's also responsible for providing the electrons found within the bond of the hydrogen-oxygen bond, covalent bond. So this is a great source of electrons. Okay. Now, carbon dioxide, on the other hand, is used to produce sugar because we're taking a whole bunch of carbon dioxide, we're piecing them together, and then creating a sugar ring. Now, energy, again, you need to inject energy when we're creating things. Some energy comes from the sunlight. So, in cellular respiration, however, it's the exact opposite. Photosynthesis is an anabolic process. Cellular respiration is a catabolic process because we are breaking down sugar into its base material. Okay. And this is just a depiction of extra complexity in the whole cycle of carbon dioxide, sunlight, energy, oxygen, etc. Okay. Oxyredox involves electron transfer. So again, one is giving, one is taking. Now, if we take a look at this second half right here, this half right here, okay, we can see that methane, if we oxidize methane, it becomes methanol. Now, why is this a process of oxidation? The reason being is if we take a look at the central atom, being carbon right here, if we take a look at carbon, right, it has four hydrogens around it. But as we oxidize, as we introduce an oxygen to the system or another component, we have to ask ourselves, the electrons around the central atom, carbon, is it going to be pulled away from the central atom to the extra thing that we're attaching it to, or is it going to stay put where carbon is? We know that oxygen itself is a highly electronegative atom. What does that mean? That means it pulls electrons towards itself. So if I introduce an oxygen to this central carbon, it's pulling electrons away from that central carbon. And if it's pulling it away, that means the central carbon is losing some of those electrons because now the electrons aren't hanging out at carbon anymore. And this is a process of oxidation. Oxidation is loss of electrons, and vice versa. If I remove the oxygen, then all the electrons are going to hang around the central atom again, carbon. Okay. So as we move down this chart, what we see is that oxygen is pulling more and more electrons away from carbon. And as we remove those groups and reintroduce hydrogen back in, then the electrons will return back to being more probable around the central atom. All right, so enzymes lowers activation energy. We know that by this point. We know that if we use the proper tools, it doesn't require that much energy. So again, think of it as traveling to Long Beach. If you didn't have a vehicle and you had to walk to Long Beach, it requires a lot of energy. Say that you live, 30 minutes away, driving. Now, if you had a vehicle and you just turned it on, you drove, it takes some energy, but it didn't take enough energy or as much energy as it would for you to wake up and walk to Long Beach. So enzymes lowers that amount of energy needed to be able to run the reaction forward. So it speeds it up by lessening the amount of energy required to run a reaction. But here's the key. Both of these processes still require some input in energy, right? To run this reaction uncatalyzed, you need this much energy up to A, right? You got to go from baseline to over that hump. You need to inject a lot of energy. With an enzyme catalytic pathway right here, you don't need as much energy, but you still need to input some energy. So always keep that in mind. Always keep that in mind. There's still certain things that we have to do. Again, with the car analogy, you still have to wake up, get into the car, and turn it on to get to Long Beach. So that still requires some energy. Now, why am I saying this? Because this next slide here is confusing to a lot of you guys. You might think this is the same graph, but it's not. Pay attention to the labeling. Remember, pictures are worth 1,000 words. If you take a look at this picture right here, you see that the y-axis is the number of molecules. The x-axis right here is the energy per molecule. Based on the axis title alone, this is all it's looking at is how much energy each molecule has and how much of that molecule do we have in the system. That's all it's saying. So if we take a look at rest, the molecule average energy is roughly around this part. Now if we say that this is 0 to, let's say, 100 kilojoules or joules per mole, right? The average amount of molecules at the substrate at a baseline at rest is maybe roughly around 10 kilojoules. Say it's 10 kilojoules. Okay. It's roughly around 10 kilojoules per mole. It's not that much. Well, it's actually quite a bit if you have 10 kilojoules. So 10 joules per mole, microjoules, whatever. It's very little. Now, to run an enzymatic reaction with the aid of an enzyme, you need to overcome that initial hump, right? You got to wake up, turn on that car, so that at this point, you've introduced or you've injected initial starting energy to run a reaction. Now, this starting energy might be 65 kilojoules per mole, whatever it might be, right? But it's that starting thing in order to run an enzymatic reaction. In order to start your car, you have to turn that key. If you don't have enough energy to turn the key, or if you're too tired to get out of bed, you're not going to ever start the car. Okay. Now, on the opposite hand, to run that same activity or the same reaction without the tool requires even more energy. Maybe this is at 90 kilojoules per mole. It's an it's an enormously higher amount of energy to run that same reaction. That's all this graph is saying. A lot of my students always get this graph confused because if I give you this graph, you're going to look at it, it's like, I have no idea what I'm reading. So again, break the graphs down and try to kind of digest what this is telling us. Okay. So with the aid of enzymes, we're able to perform the reaction many, many more times within the given time frame. All this graph is saying is that you have a reaction. It can run without it, but it's not going to perform as much as if you had the enzyme here. So if you look at the size of the arrow, you can see left side is not that much. Right side is like, okay, let's go. Let's make our product much, much more quickly. Okay. And at the start of the talk, we talked about going from A to B to C to D to E to F, right? We started with substrate A, going to F. This is another depiction of that. So we're starting with the green substrate. We got to get it to the red substrate. How does it go? By falling through these different pathways, interacting with different enzymes so that we make a final product. Now, again, it depends on what that product is, but we want to use the enzymes in a way where it's efficient. We don't want to have to create 20,000 different enzymes, right? We want to create a good 100 different enzymes. So a great way to kind of imagine the reusing of enzymes is like, I guess, a power drill, right? A power drill is a tool. The whole purpose of that power drill is to kind of, nah, that's a bad example. You know what? Let's just stop it there. We're going to keep going. If I say anymore, I might confuse everybody. So just know we need multiple enzymes to run through a specific reaction to get a specific product. Okay. And the more that we run through different pathways, the more energy it's going to take. Okay. Now, again, very specific, has to bind to the active site. So substrates are going to bind. It's going to form an enzyme substrate complex, makes the enzyme product complex, and then pop the product reusing the enzyme. Okay. Now, how do enzymes find their substrate? They don't have brains. There's nothing to kind of tell substrate, hey, come over here, I'm ready to go. It's very similar to your car. The car doesn't tell you, hey, I'm over here, we're ready to go. It's just that we have to, the substrate, we being the substrate, have to move to where the car is and sit in the car's active site, which is the driver's seat. Substrates, again, are just randomly moving through the aid of thermal motion. They're randomly moving within the cell, and eventually it's going to bind to that active site. Now, to bind to the active site, again, the aid of hydrogen bonds, van der Waals, London dispersion, electrostatic, whatever you want to think of, is going to kind of be like, oh look, I'm moving around, and then all of a sudden, boom, it attracts. So if I have something to show right here, and I'm really trying to find something right now that's metallic. Um, I have a magnet here, and I wanted to demonstrate with the magnet. Oh, right here. So right here, if you guys can see, I know my picture is very small. My little microphone here is very small. But look at this magnet. This magnet, say that this is the enzyme right here with a lot of open active sites on the plate of this magnet. Now, here's my big substrate. My microphone stand is the substrate right here. And what eventually happens is as I'm moving the microphone around, eventually what's going to happen is that it's going to get close enough to, and I wish you guys can see, it's going to get close enough to, man, we're just going to make do with it. It's going to get close enough to the actual um, active site substrate. It's going to get close enough. Here, I found a clip. It's going to get close enough to the point where, through the aid of the magnetic attraction, it's going to latch onto it. And you guys can see the magnet is latched on to the clip. Now, the thing is, I had to get into close enough proximity for it to latch on. And this is how an active site generally works. So as the substrate is moving around vigorously, it's going to eventually come within close enough proximity where a hydrogen bond is like yoink. It's going to yoink it and slide it in and fit perfectly within that active site. Now, this image right here depicts the movement of a substrate within a cell. And you can see the starting position is right here. Now it's going to move all the way around. It's going to move in a zigzag back and forth, randomly move around until it hits a final position right here. Now we can see that the final distance, the net distance that it moved, isn't that far, but it had to move through the whole entire cell. And this is within our cell. So imagine thousands and thousands and thousands of different molecules are moving around at the same time. So an enzyme has to be hyper specific because with everything moving around in that cell, it doesn't want to bind onto another substrate that it doesn't work with. It's a waste of time, waste of energy, waste of efficiency. So it has to be extremely specific for it to bind. Okay, so this is where we start talking about delta G. Know that delta G is Gibbs free energy. If it's a positive delta G, it's a non-spontaneous reaction. And if it's a negative delta G, it's a spontaneous reaction. Now, I'm not going to go through all this reading. You guys can read this on your own. You have my PowerPoint. Read it, go through the textbook, work on a couple of the practice problems, write down the equations. You will need to know it. If you are stuck on it, then you come see me. But just know, again, energy is conserved. What's being released from the system goes into the surrounding, and what's being released from the surrounding also affects what's inside the system. As long as you can kind of determine what's exothermic and endothermic, you should be able to kind of really recall this from your 111B experience. Okay? As well as the second law of thermodynamics. Entropy, again, there are equations for entropy. Look at the practice questions in your textbook. They will give you a couple so that you can try to solve it. Now, remember, when we're dealing with our R constant, most of the time, in chemistry, we use the value of 0.0802 or something like that. I forgot the R value off the top of my head. But in physics, when we're looking at moles, joules, energy, and moles, if you ever have the joules involved, okay, you have to use 8.314. I think it's 0.08206. There we go. 0.08206 is the chemistry because that's liters atmosphere per mole Kelvin. For physics or for anything dealing with joules or energy, it's 8.314. Write that down. Don't forget that. Okay. And then again, here is the different equations for Gibbs free energy right here. So you have your enthalpy, you have entropy right here. They might use slightly different variables from your chemistry textbook, but it's essentially the same equation. Okay. Energetically favorable reaction, again, delta G has to be less than zero to be spontaneous. Again, that's the only way that we can have a reaction run on its own. But if it requires a positive delta G, that means we have to input energy to keep it going. Okay. Mhm. Now, why is this important? Because every time something gives off energy, something has to also be interacting or receiving that extra energy. Now, really for efficiency's sake within the cell, we usually combine these two processes together through a relationship called coupling. So there's going to be a coupling type of reaction where one is a spontaneous giving off a lot of energy, and another one where it needs the energy to run a reaction. So it's going to couple with the ones giving off energy so it can absorb some of that energy to run. So in this picture right here, we can see the spontaneous reaction from C to D is giving out a huge, huge amount of energy. Now, because of that, some of that energy can be injected into reaction X to Y, which requires positive delta G, which is a positive delta G, which then kind of moves that reaction forward. Okay. So that extra free energy is a great thing. It helps move the reaction forward and we are being more efficient. So, analogy time, and I use a lot of analogies in my lecture. Think of it like a hybrid vehicle. We run the car on gasoline starting, so it's going to run on gasoline that exudes a lot of excess energy. But as the car is in kinetic motion, sometimes your engine's going to shut off or it's going to start turning a turbine in the back of your vehicle where it starts charging up the battery. So it's a way that we can use the excess spontaneous reaction to turn a turbine, a motor, a charger in your car itself to charge the internal batteries at the exact same time as it's running. This is what we call a coupling reaction. So that's an example. Okay. Concentration of the reactants influences the free energy and change. Yes, again, if you have more of the substrate, right? If this is Le Chatelier's principle, if you have more reactants, it wants to get to equilibrium. It's going to push the reaction forward and change the energy requirement. Okay. Equilibrium constant and delta G are readily derived from each other. Again, it's an equation. Hopefully, you guys recall that. Okay. Energy favorable reaction from concentration of Y to concentration X. The formation of X is energetically favored. So delta G Y to X is negative. Delta G X to Y is positive. Again, if the reaction is moving forward, chances are without any extra input of energy, it's a spontaneous reaction. Now, I do have to mention ice tables. Okay. Initial, change, equilibrium. Okay. This textbook, if I recall the practice problem, and I'm hoping this is the right textbook, I believe it does give you a couple of ice problems as well. So practice them. Okay. So we do talk about the constant, equilibrium constant, your K value. Remember, K value is a very simple process. So say that's X plus Y okay, yields D, okay? K value, all K value is products over reactants. And I'm trying to draw with my mouse right here. Okay, product over reactants. Okay. If your K value is bigger than the value of 1, why 1? Because if the top and the bottom are the exact same value, guys with me, if I gave product and reactant the exact same value in terms of concentration, it's going to give me a value of 1 because, numerator divided by denominator, if they're the same, they cancel each other out, we get 1. So 1 is that balancing point for a mathematical equation in a fraction. Now, if my K value is greater than the value of 1, what happens is that your products are more favored in that reaction. So your equilibrium point is on the product end of the chemical reaction. If your K value is less than 1 and close to 0, then the reactants or the substrates are favored, vice versa. So just this is what that equation is telling you. Okay. And then the free energy of X minus the free energy of Y, yada, yada, yada. Again, this is for X yielding Y. So this right here is for the chemical equation X to Y. Okay. Not X plus Y yields D. So just FYI. Okay. Free energy changes, a couple reactions are additive, so you can add more and more to it. Okay. Active carrier molecules are essential for biosynthesis. Now, we're now talking about carrier molecules, and you guys heard this all the time when you're doing photosynthesis and cellular respiration. We have carrier molecules that carry electrons, your electron taxis, your electron bus, your electron transportation, whatever you want to call it. We have those. Now, remember, as we're releasing a lot of energy, electrons, biochemical energy, chemical reaction, electrons are being transferred. Now, there are certain times where we can couple that because we don't have another reaction to couple it with. So we are going to charge a battery so that we can use it at another point in time, just like your hybrid car. You're going to charge your internal battery. So in the event that gas runs out in your hybrid, you're able to run on electricity until you get to a gas station. Okay. So this is just an example of where we can couple the energy, and sometimes we just don't have another reaction to couple it with. So we're going to bring in an electron carrier to kind of absorb that extra energy, use it for later elsewhere. Okay. So energy, again, with the catabolic process from food to basic building material, energetically favorable reaction. Again, breaking anything down is much easier than building things up. Okay, so this is an exergonic reaction or exothermic reaction, either one. Exergonic is mainly for biology, though, in my opinion. And as we do that, we have our electron carriers. Now, what are the electron carriers? FAD, right? NADH or NAD plus. NAD plus, FADH plus, something like that. Okay. I'm blanking right now. I haven't had coffee yet. All right. So, and it's going to go to FADH2. Oh my gosh, drawing with the mouse sucks. And NADH. Oh my gosh, what is going on? Um, oh gosh. Okay, there we go. Woo, mouse. So those are our electron carriers. Okay. And again, it's just so that we can use it in a later sense. Now, ATP also is an energy storage molecule. Very helpful. Remember, the energy is found within the bonds themselves. Now, take a look. Take a look at this. Why is there so much energy between two phosphate groups? Why? Well, take a look at the central atom, phosphorus. All the electrons are being pulled away. right? You guys agree. There's so many oxygens around phosphorus that all of it is going to get pulled away out of phosphorus. So phosphorus is extremely positive at this point based on the chemical structure of or the atomic structure of each element. Okay. Now, oxygen being an electronegative atom is also surrounded by other oxygens. So in between this bond, you have a lot of electrons as well as a lot of electrons elsewhere that's surrounding that bond. So when we snap it, when we break it apart, it's going to spring load out. It's going to just break, releasing a huge, huge amount of energy. So ATP, ADP, CTP, CDP, GTP, GDP, those are all energy molecules that cells use. ATP being the most common and probably the universal one for all life on Earth. Okay. Again, hydroxyl group on another molecule breaks it apart. Okay. Forming a activated molecule because again, phosphate needs to be attached to activate that molecule. And so we go from an ATP, we break that bond, we attach it, couple it to another molecule because it's a coupling reaction. We activate that molecule, and then ADP is left. And hopefully, we're able to break that inorganic phosphate apart later on in a different reaction, break that phosphate group out, and then reattach it to ADP once more to make ATP again through ATP synthase. Okay. Okay. Again, we use it to join molecules together. It is an energy intensive process. So ATP is going to be one of those main sources. Now, NADH and FADH2 are electron carriers that can also inject energy, but mainly in the citric acid cycle. Okay. Or mainly produced in the citric acid cycle, injected into an electron transport chain. Yep. NADH and NADPH are also. And then here's an example of a cholesterol. Okay. Dehydrocholesterol. So on the seventh carbon, there isn't an extra hydrogen right here. So if you take a look at these carbons right here, it's no extra hydrogen. However, with the aid of NADPH and the hydrogen gradient, we can break that double bond, adding and fully saturating the carbon so that we change the chemical structure from 7-dehydrocholesterol to regular cholesterol. Okay. Now, why does it break right here? Well, again, this is an organic chemistry concept, which we are not an organic chemistry class, so I'm not going to ask you guys that. But just know oxidizing agent for catabolism, reducing agent for anabolism, oxidizing agent, whenever the word agent is involved, oxidizing agent, it is an extra molecule that helps in a reduction process. Okay. So whenever you hear oxidizing agent or reducing agent, just think opposite. Reducing agent aids in the oxidizing process, and oxidizing agent aids in the reduction process. And many other active, yep, coenzyme A right here is your coenzyme A, right? So acetyl group, again, when we break our pyruvate, the three carbon molecule, we get rid of that extra carbon and attach a coenzyme. It's an acetyl coenzyme or acetyl CoA. Okay. We have FADH2, again, two hydrogens right here. Okay. We have, yeah, two hydrogens. And then here are the rest of the extra, what do you call it? Electron carriers. Okay. Or energy storage molecules, whatever you want to call them. Okay. And this is just, again, giving you an example of different biochemical process. We're in chapter 2 right now. We will talk about this, I believe, in chapter 5 or 6. But yeah, synthesis of ATP hydrolysis, the synthesis of biological polymers in ATP hydrolysis. Again, hydrolysis, breaking using water. Okay. So we're using water, breaking ATP apart, and with the extra phosphate energy that just got broken, we synthesize things together. Okay. Yep. And here's an example of how we use water to break apart ATP. So we can go from ATP to ADP and then ADP to AMP, adenosine monophosphate, which is a precursor to certain signaling um, molecules. Okay. Cyclic AMP more so is the signaling molecule. Okay. Again, this is again showing you guys building and breaking of polymers. Okay. Glycolysis is the central ATP producing pathway. Again, this is just a review of cellular respiration. Okay, you got to go understand fermentation in the absence of oxygen. Oxygen is required for cellular respiration, the efficient form of cellular respiration to occur. Remember, oxygen is the last electron acceptor in the electron transport chain. Without the electron acceptor, the electron transport chain can no longer function, hence why it undergoes a fermentation or alcoholic pathway. Okay. And you guys can see the different types of energy. So this is enzymatic energy without this is just direct burning of sugar increase and then boom, a lot of energy. But for our body, because we use multiple enzymes, it's multiple humps to get to carbon dioxide and water. Okay. Uh-huh. And again, this is just an introduction of glycolysis. There are 10 steps in glycolysis. Okay, so step one, step two, three, four, five, six, seven, eight, nine, and 10. We're not going to go too heavy into this right now. We will talk about it as we go forward, but this is mainly a biochemistry process, which will be more detailed as we go through the semester. Okay. And then again, this is just building the aldehyde here. Okay. Using coupling reaction gives free energy for each of the changes in the molecular structure. So of the pyruvate as it's breaking. Okay. Fermentation in the absence of alcohol. Again, this is demonstrating how NADH is being used or FADH, mainly NADH, not FADH. What am I talking about? NADH used in the fermentation process where it's coupling from pyruvate and conversion into a lactate or ethanol. Okay. And organisms store food molecules in special reservoir. Again, they need to if they want to survive. A lot of our muscle cells store glycogen as a source of a quick energy source for food. So just keep that in mind. Glycogen, glycogen polymers, yes. So carbo load before doing a high intense endurance workout helps. Okay, we want to fully saturate your muscle cells so that it has food to run. Okay. Fat droplets, again, cell wall plasma membrane, 1 micrometer big. Okay. It's not nanometers, it's micrometers. Okay. Different storage food right here. Animal cells derive their energy from fatty acids obtained by fat. Yes. So, again, why we store our energy as fat is because it is a great energy storage. Now, remember, to go from in cellular respiration, there's three steps. There's glycolysis. Okay. Now, remember, what's the point of glycolysis? It's to convert C6H12O6 glucose into a three carbon molecule called pyruvate. And then to have that pyruvate function in the cellular respiration process with the presence of oxygen, we're converting pyruvate into acetyl, which is a two carbon molecule attached to a coenzyme A. Now, our fatty acids are long chains of hydrocarbons. Oh, look at that. And we have a process called beta oxidation where we take two carbons off of the chain each time, attach an acetyl CoA to it, and inject it into the mitochondria to perform cellular respiration. Hence why burning of fat is better in a aerobic exercise versus an anaerobic exercise. Okay, so if you really want to burn fat, you have to kind of nudge, the beta oxidation process so that it can go through this. Now, remember, there's other ways that we can digest energy or form energy from other macromolecules. So carbohydrates being the main source. But if you don't have carbohydrates, proteins is another source that we can break apart because remember, there's a nitrogen carbon carbon group. So NCC, there's still two carbons in there that we can still digest. Okay. And fatty acids. Fatty acids being the hardest to digest because that's usually used only in case of an emergency. And there's a whole health trend, with ketosis, carnivore diet, etc., that, not introduces, but it promotes this type of oxidative process. Okay. So again, sugar and fats right into acetyl-CoA. It breaks down into acetyl-CoA, making it available to run into the cellular respiration process. Okay. So citric acid cycle. So once you have acetyl-CoA, you can run through the 8 steps of citric acid cycle, and then out pops electron energy. Okay. GDP, GTP, again, be familiar with different energy carriers. Okay. Okay. And again, alpha ketoglutarate, yeah, isocitrate. Again, these are all the extra steps within the citric acid cycle. Okay. Electron transport drives the synthesis as always. So we have glucose going into the citric acid cycle through pyruvate and acetylation, runs through citrate, alpha ketoglutarate, succinyl-CoA, oxaloacetate going back into citrate. It's all conversion of carbons. So are we breaking carbons apart? Are we putting them together? What are we doing? So it goes from a 4 carbon molecule going all the way around back to a 6 carbon, back to a 4 carbon, et cetera, et cetera, et cetera. Okay. Okay. Now, do take note. Certain products in the citric acid cycle is also can be exported out, uh, making cholesterol and fatty acids. Okay. Oxaloacetate is used to make aspartate. Okay. Succinyl-CoA is used to make your heme molecules, blood. So a lot of these can get involved with other biochemical processes. Okay. Animals must obtain nutrients from nitrogen and sulfur. Okay. So here are your essential amino acids that we need to get from our diet. This is why we're called essential because we don't create these on our own. We need sulfur, we need nitrogen to be able to make our different molecules, mainly our amino acids as well as our nucleic acids. Highly organized and regulated. Obviously, it's going to be regulated. You need to have your metabolism regulated. Otherwise, you're either going to consume too much food, energy, which will eventually digest, you're going to eat from the inside out, or it's not going to, it's producing too much storage where it kind of bloats out. So, for a typical cell, it's going to be extremely regulated so that it has a balanced flow. Okay. And we can see just in the or the glycolysis alone, this process right here, look at how it's embedded through all other process within the cell. The red path, minus my drawing, is what is a small, small biochemical process. And if you guys ever take 441A, you'll go into this into super, super detail, but you're also going to start learning about all the other processes. It's incredibly intricate and it just shows how chaotic our cells are, but in this chaos, how organized and regulated and methodic the cell is to be able to kind of function in that chaos. It's a beautiful thing. And so that's it. I hope I didn't lecture too long. I really hoping that this is less than 45 minutes. So again, I sped through that last half. I recommend reading the textbook on the parts that I sped through. Citric acid cycle glycolysis, yes, you'll need to know the steps of that, but I'm not going to be asking anything in terms of memorizing exactly what's happening, mainly the overall process. Why is it that, these steps are required? Where is energy being used? Where is energy being created? Those are the things that I do want you guys to have a good reference point to. In terms of Gibbs free energy and all of that, please practice on a couple problems. You work with the equation just to refamiliarize yourself. I'm pretty sure that most of you guys just need one or two problems to kind of remember how to do it, and then you should be good. Other than that, be on the lookout for the next several videos. I don't know when they're going to be posted, but they should be posted very soon. And hopefully, this was a good part two for you guys. And until then, again, keep working on practice problems. Come to class, ask questions if needed. And yeah, see you guys in the next video.